Hello students. Okay, so in this lecture we will be discussing a really important topic for your NEET examination point of view. Okay, so can anyone tell if a red flower and a white flower are crossed, why do we sometimes get a pink flower? And in humans, how can someone have both A B blood groups? Yes, there are AB blood groups, right? Some people have AB blood group, AB positive, AB negative. Today we will understand two important NEET concepts. One is incomplete dominance, okay. Another is codominance. And both of them are exceptions to Mendelian concept. Clear? Okay. So, firstly, you must watch the lecture of Mendelian cross, the monohybrid cross. Also, you will have to know about the dihybrid cross. Clear? What the laws were. So, I will just give a basic recap where Mendel told one allele will be dominant. Okay. Suppose capital T and small t. Capital T is dominant over small t. That's why we are getting the tall phenotype. The organisms, the plants become tall. Okay. And the capital T is a dominant allele. And the small t is a recessive allele. Right? But this was the thing we learned. But in some cases, this dominance does not work. Okay? The dominance does not work. So, what happens in this case? Let's start. Now, what happens is, in case of incomplete dominance, I will be telling first about the incomplete dominance. Okay. Okay. So, what the definition tells is, in this, neither allele is completely dominant. Okay. So, the phenotype shows intermediate. What does that mean? We will be learning with an example. There is a flower named, the example we are taking of a flower. Remember, Mirabilis jalapa. Clear? So, when we cross the red Okay, this is the red flower of Mirabilis jalapa, and this is the white flower of Mirabilis jalapa. Clear? What happens when we cross it? Suppose this is capital R, and this is small r. Let's do the crossing work. Capital R, and small r. So what will be the gametes? Capital R and small r. Okay. So we were telling that none of it was dominant. Means not this R, this capital R, not this small r. None were the dominant alleles. So what will happen is it will produce a phenotype or a heterozygote of capital R or small r. Okay. Where none of them are dominant over each other. So what it will produce? See, the genotype will be pink. Clear? This was red, this was white. As none was dominant, this will create a pink phenotype of flowers. And you can easily tell, if R was dominant, what would happen? If R was dominant, it would have created red flower. Clear? And if small r, if capital R was dominant, it would have created red flowers. But if small r was dominant, it would have created white flowers. Clear? But in this case, none of them are dominant over each other, none of the alleles. So what will happen? It will show incomplete dominance. Okay. So pink color phenotype of flowers will be produced. Clear? So now you will have to tell me what will be the phenotypic ratio. Okay. When you do the whole cross, you will get the phenotypic ratio of 1:2:1. Okay. Suppose you will have to self cross it. Upon self crossing, what happens? See, capital R, small r, okay, capital R and small r are the gametes, and this is capital R and small r again. So capital R, capital R, small r, capital R, small r, small r, small r. Okay. So the thing is, this capital R will produce red flowers. Clear? Capital R, small r will produce pink flowers. Right? And small r will produce white flowers. So the phenotypic ratio is, this will be 1, this will be 2, and this will be 1 again. So that will be 1:2:1. But in case of Mendel, okay, in Mendel's monohybrid cross, what was the ratio? phenotypic ratio was 3:1. That's because this capital R or capital T was dominant over small t. That's why both of these were in tall category. But in this case, there is no dominance. Clear? This is about the incomplete dominance. Now, let's talk about the codominance. So, the definition first. Both alleles express simultaneously. Okay. So, both the alleles will express simultaneously. Clear? So, the example is ABO blood grouping system. Clear? Okay. Now, let's talk about the alleles. From this, we will be getting three types of alleles. What are the alleles? The alleles will be IA, IB, and only small I. Okay. See, these are the alleles. Okay. Now, let's see what happens in the case of codominance. Clear? Suppose you are crossing IA and IB. What you will be getting? See, you will be getting IAB. That is the AB blood group. Clear? So, both the alleles will be expressed. Okay, both the alleles will be expressed as both of them are codominant to each other. But this I allele, small I allele, is recessive one. Okay. This will be only in the case of AB blood groups, people having the AB blood groups. So, you can just see this white cow, and suppose you make it a black cow. Okay. Suppose this is a black cow. Okay. I think you will be able to understand with it. This is a black cow and white cow. What happens when we mix both of them? What we will be getting? We will be getting a phenotype of cow which will have, in white it will have spots of black. Clear? Spots of black. Okay, like this. So this is codominance. This is codominance, where both white and black present. Plus means present. Clear? But in case of incomplete dominance, what happens with white? Suppose you are crossing the black one, what will happen? This cow which will be produced will be gray in color. Clear? So no white, no black. I think you have understood very clearly. Incomplete dominance no white, no black, gray is produced. So, no alleles or genotypes are produced in the phenotype. Okay. So thank you for watching. If you want these notes, you can contact me. My contact number I am giving it, sharing it. It is 89108 77749. So thank you for watching.